So we're going to move to our next speaker, who's Dr. Shoemaker, who's going to talk about metabolomics profiling of brain injury.
Well, Mr. Dryden, thank you for that, and I regret to inform all of you now that this is not about prevention. Perhaps not even secondary prevention. I'm Kevin Shoemaker. I want to pass along regrets from Dr. Doug Fraser, who can't be here for family reasons. As I found out about 6 days ago, I would be happy to give a talk about something I know very little about. He's a biochemist, I'm a physiologist, and so he's been working in the area of metabolomics for a while, and I for about 6 days. So I'm going to do my best to deliver what I understood of his talk. I removed the slides I didn't understand, except for maybe 2, and you'll catch on which ones those are. And then I've supplemented that with some of the work that we're doing with Doug, and it's a bit of a self-promotion thing here, okay? So I apologize for that as well, but it reflects on some of the work that's being presented here by students out of our lab and our combined program. So if you're okay with that, I'll do my best. I can't give you the same information that Doug would. I hope I can replicate his passion for what he's doing, and which I think is really cool, and I'm very proud to be part of his team and to work with him on this. We're speaking about mild traumatic brain injury here, so concussion, and particularly focused on adolescents primarily. The problem with a mild concussion, compared to the more severe injuries I think that we've been speaking to so far, no obvious external injuries occur. Clinicians are generally poor at recognizing a concussion, partly because concussion symptoms can occur in those without a brain injury, particularly adolescents who tend to be moody at various times. Headaches come and go. You know, there's a variety of symptoms that show up that aren't part of a head injury. And commonly available imaging techniques, such as CT scans, are not good at detecting concussion. They're not good at detecting damage, although we think it's there because of the behaviors that we see. So one of the tasks that we as a team have been working on has been to identify novel biomarkers for concussion diagnosis and also for the management. And Doug's group has been working on metabolomics in particular. Now, before I describe what metabolomics are, what is a biomarker? A biomarker, to be valuable, has to be sensitive, specific, and reproducible. It has to reflect the nature of the injury, the degree of the injury, the time course of the injury. It has to be affordable, widely available, standardized across institutions, and ultimately linked to a clinical decision. Currently, we have no such biomarker. This biomarker needs to inform diagnostics. It has to be able to tell you something about the time course of the injury. Is this going to proceed to post-concussion problems, or is it going to resolve in a week? That'd be nice to know, because that would then inform the therapeutics of this injury, we hope, and it would also help us understand what is the best therapy for this. Should we include exercise or rest, for example? That's the hot topic right now. And ultimately, are they okay to go back to school? Are they okay to go back to play or to work? Something to help that clinical decision would be, beneficial for sure, and we're not there yet. The sources of biomarkers have been plentiful. The fluid biomarkers are probably the most studied. We also have behavioral ones, which I'll get to in a moment. Fluid biomarkers, cerebrospinal fluid or blood plasma are the primary sources. And you've heard about some of these already, but because it's related to the brain and there's this idea of a metabolic problem, there's a vascular problem, there's neuronal damage, all of the things listed on this slide reflect markers either from neural damage, axonal damage. You've heard a lot about tau. You have markers that reflect astrocytes and glial cell damage and neuroinflammation. These little cells are very important. I'll be touching more on them later. And there's also markers around the endothelial layer of blood vessels, for example, that blood-brain barrier that's so important to protecting the brain from subsequent injury. Lots of these have been looked for and sought after, primarily in more severe trauma. In the area of mild traumatic brain injury, however, none of them have panned out yet to be particularly helpful, although I would say we're still in the very early stages of this research. Not to discount the problem, though, that the game is played tomorrow. Metabolomics is the systematic study of a unique chemical fingerprint that specific cellular processes leave behind. So it's the exhaust after the bus has left. But that tells you the bus has left, and it tells you something about the engine, in that the exhaust should smell a different way than it does. Or it can tell you it's a diesel engine or not, for example. So metabolomics are about metabolites. It's not about genomics or proteomics. It's about what's left behind as the cell does its work. When you look for this stuff, why am I shining at that one? It's this one. I just happened to look there. Sorry. If I hitch anybody's eyes with this thing, be careful. Okay. So you use mass spectrometry, spectroscopy, nuclear magnetic resonance imaging, and spectroscopy primarily for this work. And you basically take a blood sample because CSF is not available in mild traumatic brain injury. And you look for metabolites of various processes. Amino acids, isocarnitines, glycerophospholipids, sphingolipids, and sugars are primary targets. And within each one of these, there's multiple metabolites that combine into these families. Oops, sorry. So in the preliminary study that Doug's team was leading on, and it's now, it has just come out, actually, just published, the data were based on male hockey players, 12 to 14 years of age. They had not had a concussion in the previous 6 months until now they are a patient, and within 3 days of their injury, the blood sample was obtained. So relatively small N size, which I think you need to put in perspective how powerful this method may be, because you can use it on small N sizes, and the idea is to get it down to, can we tell if you have a concussion? Not a group of 12 or 15 or 40. Okay, it's important to be able to study individual people. So these data come from 12 concussed patients and 17 controls, all the same age, hockey players, each one of them. 2 to 3 days from the time of injury to the blood draw. Total symptom score on the SCAT-3 was 12 in terms of symptom severity, and 29, severity was 29 and symptom score was 12. So definitely fall into this moderate brain injury category. So analyzing a blood sample with mass spectroscopy or NMR is one thing. You get a list, in our case, of about 174 metabolites. You have to make sense of that, and the way you make sense of 140 variables changing all at the same time is you take it to a mathematician, and they run their analytical informatics modeling and predictive modeling, machine learning, all these things that I don't understand. But they do. But here's the process, and the idea is to whittle it down to something that's understandable. And that's why all the processing has to occur. Pre-processing is simply getting your samples. You can sort of list them and put them in a spreadsheet. Principal component analysis is one way to understand the fundamental data and how many, what they call dimensions, which have to do with the number of amino acid families and metabolite families. You can further then get rid of features that you don't like using these kinds of modeling here, distributed stochastic neighbor embedding. I can read that. I can't tell you how it works, but it's nonlinear compared to principal component, which is, and so forth. You eventually get a data that you can start clustering and understanding which groups fall together. You can then take all of these and mix it all up and say, well, can we predict using a machine learning model, like basically run through the data once, get rid of one, do it again, and see which one of these multiple metabolites actually predict if you have a concussion, and where do you sit on the charts. This is the principal component analysis, sort of the first phase of the analysis. All the data are normalized, so these values become unitless, essentially. But what we have is two groups, and you can clearly see them. We have a red group, and the dotted line is the 95% confident interval. We have a green group, and I hope you can see that. The green group is the controls, and the red are the concussed. And because those are the 95% confidence intervals, you can see there's nice separation between the two groups. And not any of them overlap into there, as a rule. There's different ways to analyze these data then as you try to streamline down to the important elements of this. And I'm just going to blitz through these slides to tell you that you can look at these different ways. Here's a hierarchical clustering, for example, where you've got concussed in the green. This is not going to show up very well in the room. Sorry, concussed in the red here. The green is concussed, and then you mix them up as another batch. And then you look for visual presentation. All of the whites are essentially people that are very, very different. All of the reds are values that are very much the same. And clearly, the concussed do not look like the mixed very well, or particularly the concussed do not look at all like the non-concussed. And so that's a visual representation of the kinds of analytics and multivariate statistics that go on behind this onto something we understand. Okay. Here's a list of the metabolites that are the most predictive of a concussion. So the ones on the left have achieved 92% accuracy in predicting someone with a concussion versus not. And that's based on a chi-square analysis, and there's 20 metabolites are listed there. On your right of the screen, there's 40 metabolites listed. They've added a few more components, and you can get the accuracy up to 95% from 92%, and that's using a slightly different analytical process. But the point is, 92 to 95% accuracy is actually, when you're trying to find a biomarker, that's actually kind of phenomenal for a clinical biomarker to be that accurate. The SCAT, by the way, is around 80%, as I understand it. And that is the only clinical tool that's used, actually, to corroborate the intuition of the clinician. Now, so let's just zoom in on the most important data that actually produce the highest predictive value. Look at the names of these components. They are almost all the same. They're just slightly different. And what these represent are glycerophospholipids. The glycerophospholipids family of metabolites comprises 82% of all of this predictive ability. It's that family that seems to be very important. What's really fascinating here, though, is kind of counterintuitive to me. I think a damage you would expect to see more of something, but in this case, we're seeing less glycerophospholipids than more. What is a glycerophospholipid? It is part of your membrane. It's very important in neural tissues, in astrocytes, in any kind of tissue, essentially. They are dynamically regulated. You think about all of the metabolic processes that happen inside a cell. They involve usually some lipid bilayer breakdown, so they're involved in that kind of thing. They're involved in protein structure, protein stability within the membrane. And so they're very important in anything that happens on a brain. They account for 25% of the adult brain dry weight. Myelin contains the highest content in the brain, and as I said, they're dynamically regulated. So what happens in a damaged brain? Should they go up? Should they go down? Would they be present at all? In our analysis here, they seem to be very important in predicting that damage has occurred, except the levels went down, and I have no idea what that means. So to quickly summarize this, metabolomics profiling of a concussion using blood metabolomics profiling and math and machine learning, we've identified concussed individuals with 92 to 95% certainty. That's pretty good. In fact, it's clinically outstanding. Much of the observed variance between groups was due to changes in plasma glycerophospholipids. So this is like a data search, right? We didn't start with a hypothesis on this. We just had a bunch of data, we dumped it in, we did some math, and this comes out. And so now we can generate this hypothesis that there's something cool happening with glycerophospholipids in particular, and we think it's relevant to the brain. And so that gives us a place to start searching in a very directed manner. So we're kind of excited about that. And will this turn into a diagnostic? Well, we have to go back to the criteria of what a biomarker is and fulfill all of those criteria first before we can say that. But this is an excellent start and a place to start, we think. Okay, so what? This is where we get into the part of the group efforts that I'm involved in. Ultimately, neural damage, if it has a metabolic signature that we can detect, does that signature then relate in any way to the vascular and neural components that are also associated with a concussion? And if you read all certainly the severe concussion literature, the idea is that there is a metabolic insult, a vascular component to that that is associated in some way with neural misbehavior of sorts. So I'm not going to steal the thunder of my students here. I'm actually going to present and tease you a little bit, at least I hope it does, on some of the things that we're trying to do with this. Let's begin with the vascular part. In the brain, you have neurovascular units, you have nerves which communicate with each other. The astrocytes, which are star-shaped glial cells, are linking the neural behavior to the vascular system. In fact, the end feet of these astrocytes cover about 95 to 99% of all the capillary and arterioles in the brain. It's the astrocytes that do the work, and they allow the communication between nerves and blood vessels to work properly. There's a very interesting study that Christine Augustine did with Doug Fraser and Gediminas Sapinskas in a tissue co-culture model using human cerebrovascular endothelial cells and human astrocytes, and you put them together with a small perforated glass plate in between. They grow, the astrocyte feet grow, and they latch onto these endothelial cells, and you form this kind of a structure. The model of trauma here was an air puff on that preparation, which to a cell is damaging. And what happened was these end feet pulled away from the blood vessels, or from the endothelial cells. That should affect neurovascular control. It should affect endothelial function. And so we wanted to take that to a human model to some extent, at least look for it. And the problem in any human studies when you're looking for behavioral outcomes, neural outcomes, is I think, this is my personal bias, but measuring something at rest doesn't tell you how homeostatic physiology works. Okay? It works by being stressed and reacting to it. And so we've taken that approach to the study. Brain blood flow is very well regulated under baseline conditions, and it's very difficult to find, at least on a global sense, impaired blood flow in a concussed brain. MRI imaging systems are finding little pockets of damage right now, and that's sort of state of the art, and it's moving forward. Well, to avoid steady state conditions, we're taking advantage of the high temporal resolution of Doppler ultrasound. And here's something that a concussed person does a lot in a day, and that's simply stand up. And when you stand up, blood pressure here falls very much, about 20 millimeters of mercury, very quickly. Concurrently, blood flow in the brain also falls. But then it becomes restored. And the trick in the brain is to restore blood flow to the brain before blood pressure comes back up. That's what autoregulation does in the brain. It tries to preserve brain blood flow. You can mess with that and calculate the rate of dilation of the brain, which is an index of vascular function. And I'll give you some data here. Aaron is going to present this shortly. But just showing you that this slope of this drop in vascular resistance, called the rate of regulation here, is lower. This is in a small group of concussed individuals compared to controls, whereas baseline cerebral blood flow appeared to be the same. Neural function. I'm an autonomic nervous system guy. I like physiology, I like homeostasis, and so I study the autonomic nervous system. It's also inherent in any kind of physiological response. We've also been studying the brain a lot and trying to understand autonomic function in the brain. Those of you who study things like the default mode network or do neuroimaging, these regions should look very common to you. And I know there's the MRI data is teasing us right now to say that in a concussion, there's something different about the default mode network. That's the network that's you're daydreaming. That's the one that's active. As soon as you form your attention to something, it becomes less active, that kind of thing. And it's well distributed. But these sites of the brain are involved, we believe, over a number of studies, and we're not just the only ones, are involved in the autonomic control of the heart. And you can measure that, and these are very similar to the default mode network, by the way, but just blitzing through here, you can measure that, at least in a robust, you can get lots of data, but it's sort of hard to interpret, but heart rate variability, for example. just the variations in time between each R wave on an ECG recording. We've been doing a lot of study now on adolescents, and it's simple data to collect. I'm going to show you data when they're lying down supine, and draw attention to the difficulty of trying to assess this stuff in people while they're lying supine. So Chris is going to present this shortly, and we have a poster out there too showing you some of the more dynamic properties of heart rate in concussed. But this is based on 30 controls now and 30 concussed individuals. And here, when you do heart rate variability, I think there's 23 different ways of assessing heart rate variability. We're listing 7 or 8 here. And some of them, I'm not going to describe any of them here, but some of them show a difference between the two groups using straight up t-tests, whereas others do not. The question becomes which ones are meaningful, and what do you look for here? Do you look for a pattern? Do you want 23 out of 23 or 18 out of 23? We don't know. The problem is it's not very compelling when you just look at supine data. But Chris is going to show us what happens when you make them do something, and as is Emily's poster. And then you start seeing some dispersion between the two groups, very clear dispersion. With that, I'm going to stop and thank you for your attention. And if there's time, I'll address questions. And if not, I understand. We're going to... Okay, I got to go. I'll see you.
We'll refer you to the posters.